Over an aerial view of a stone monument, overlooking the city and the Life Sciences building, text reads, University of Dundee, School of Life Sciences. Using microscopes to study Parkinson's diseases. A montage shows scientists working in labs, reviewing data and preparing samples.
Our laboratory in the Medical Research Council Protein Phosphorylation and Ubiquitylation Unit, or MRCPPU for short, studies how cells recycle mitochondria by a process called mitophagy.
Two microscopic images of tissue. On the left, a black worm-like shape with thin white lines is circled in white and labeled healthy mitochondria. On the right, is a black oblong shape with uneven outline, wrapped in a gray outer layer. This is circled and labeled old mitochondria being recycled.
Mitochondria, the cell's power stations, must be recycled when they get old. A failure to do this in the nervous system, specifically neurons in the brain, can lead to neurodegenerative diseases, such as Parkinson's disease.
Microscopic footage of mitochondria shows a network of green and yellow lines and bright red dots against a dark background.
We have developed a stain, shown here in this video, of live human cells, which allows us to measure mitophagy. Healthy mitochondria have greenish yellow colour, while those being recycled are red. For scale, the healthy mitochondria appear as worm-like structures and are approximately 5 millionths of a metre long. The recycling mitochondria in red are much smaller as they have been broken up into smaller pieces for recycling.
In a lab, a scientist places a vial on a desk. He sits down and removes the lid of a box containing dry ice. The vial contains a clear liquid and a small, beige brain.
We think that boosting mitophagy in the neurons of people with Parkinson's disease may be a way to treat the disease. To see if this is possible, we need to be able to watch this process happening in these neurons. We use mouse brains that have our special mitochondrial stain to study this. First, we need to make very thin sections of the brain using a sledge microtome, which is somewhat like a mandolin slicer used to cut food.
The scientist fills the tray of a sledge microtome with chunks of dry ice, then pipettes a solution onto a ribbed plate surrounded by the dry ice. The scientist uses tweezers to place the brain atop the now frozen cloudy solution, and then pipettes a few more drops of solution around the brain.
The brain needs to be very cold to make thin sections, so we place the sample on a stage cooled with dry ice at -78.5 degrees Celsius. And fix it in place with a sugar solution.
The scientist slides the blade of the sledge microtome towards the brain and cuts a thin slice.
Once the brain is frozen, we cut 50 micron thick sections, which is about the thickness of a human hair.
The scientist collects the thin slice of brain tissue from the blade. The scientist submerges the thin slice of brain tissue into a clear liquid solution in a well of a 24-well plate. The other wells also contain slices of brain tissue submerged in the clear liquid solution. The scientist pipettes the liquid from the wells and replaces it with a detergent. The plate is then placed on a rocker.
We place these sections in a plate containing PBS, a physiological salt solution, and stain them, which takes a series of steps. We remove the PBS from the plate, add a detergent, and place on a rocker. Small holes are made in the brain sections by the detergent.
The scientist extracts the detergent, replaces it with an antibody solution, and then places the plate back on the rocker.
After 15 minutes, we remove the detergent and add an antibody. The antibody can get into the brain tissue through the small holes and mark the dopaminergic neurons. It is these neurons that die in Parkinson's disease. We place the plate on the rocker overnight.
The scientist removes and replaces the antibody solution, and then places the plate back on the rocker.
The next day, we remove the antibody and add a second, which includes a blue dye that will let us see these neurons under the microscope. We place the plate on the rocker for 1 hour so that the second antibody can stain the first.
The scientist takes a slice of brain tissue, washes it in a clear solution, and gently lays it on a microscope slide. He drops mounting liquid around the tissues, and then seals a cover slip in place with clear nail polish. The slide is added to a collection of slides inside a red box.
Finally, we mount the brain tissue on microscope slides. To do this, we select a piece of tissue, rinse it in PBS, and place it on a slide. Mounting liquid is added, a cover slip is placed on top, and its edges are sealed with clear nail polish. We can then go to our special high-resolution microscope to take pictures.
The scientist adds a slide to the microscope, and then looks through the eyepiece. At a computer, a scientist looks at an image captured of the slide. A 3D image rotates on the screen. It has an uneven surface colored blue with small red dots, some individual and some grouped together, sparsely distributed above the blue surface.
After loading the slides, we locate the area of interest and take a picture. Here we can see mitophagy occurring in these specific neurons. We can also reconstruct the images in 3 dimensions to visualize the areas where mitophagy is happening.
Scientists in a lab discuss and take notes. In a meeting room, a scientist presents a slide with text reading, mitophagy in the brain, DA and Purkinje neurons. Mitophagy impairment in DA neurons. No effect in Purkinje neurons.
This research is allowing us to determine when and where mitophagy goes wrong in diseases such as Parkinson's. This will allow us to find potential treatments that can restore or improve mitophagy and hopefully aid in finding a cure for this debilitating disease.
Over a white background, a black letter W appears over text to form the Wellcome logo. Text below reads, supported by the Wellcome Institutional Strategic Support Fund awarded to University of Dundee. A black and blue symbol appears with the letters UKRI emblazoned over it. Text reads, MRC protein phosphorylation and ubiquitylation unit. A blue and white crest appears beside blue text to form the University of Dundee logo.